morning, viewer. Welcome to our Glory Globe TV's amazing news for the viewer. Researchers at Baylor College of Medicine have identified a natural process in the brain that can remove existing amyloid plaques in mouse models of Alzheimer's diseases and help maintain memory and thinking ability. This process relies on astrocytes, star-shaped brain cells that can clear away the toxic plaques commonly found in Alzheimer's. When the researchers increased productions of SOX9, a protein that guides many astrocytes' function as the brain ages, these cells became more effective at eliminating amyloid buildup. The findings published today in Nature Neuroscience point to the possibility of developing treatments that strengthen astrocytes' activity to slow cognitive decline in neurodegenerative condition. Viewers, astrocytes. Astrocytes perform diverse tasks that are essential for normal brain function, including facilitating brain communications and memory storages. As the brain ages, astrocytes show profound functional alternation. However, the role these alterations play in aging and neurodegeneration is not yet understood, said first author Dr. Dong Zhu Sui, who was at the Center for Cell and Gene Therapy and the Department of Neurosurgery at Baylor, while us working on this project. Choi currently is an assistant professor at the Center for Neuroimmunology and Glial Biology 2, Institute of Molecular Medicine at the University of Texas Health Science Center at Houston. In this study, the team set out to uncover how aging affects astrocytes and how those changes relate to Alzheimer's disease. They focused on SOX9 because it acts as a key regulator for many genes in aging astrocytes. We manipulated the expression of the SOX9 gene to assess its role in maintaining astrocytes function in the aging brain and in Alzheimer's disease models, said corresponding author Dr. Benjamin Dandin, Professor and Dr. Russell J. and Marian K. Blanchard, chair in the Department of Neuroscience, director of the Center for Cancer Neuroscience, a member of the Duncan Comprehensive Cancer Center at Baylor, and a principal investigator at the John and Dan Duncan Neurological Research Institute at Texas Children's Hospital. An important point of our experiment design is to work out the mouse models of Alzheimer's disease that had already developed cognitive impairments such as memory deficits and had amyloid plaques in the brain. Choi said, and we believe these models are more relevant to what we see in many patients with Alzheimer's disease symptoms than other models in which these types of experiments are conducted before the plaques form. In this Alzheimer's model, the researchers either increased or removed SOX9 and then monitored the cognitive abilities of individual mice for 6 months, tracking how well these recognized familiar objects or environment. When the study conducted, the team examined the animals' brains to evaluate the amounts of plaque that had accumulated. Compared to reducing SOX9 expression, increasing it had the opposite effect. SOX9 knockout accelerated plaque formation, reduced astrocytes complexity and decreased clearance of amyloid deposits. Overexpression reversed these trends, prompting plaque clearance while increasing the cell's activity and complexity too. Importantly, overexpression of SOX9 also preserved cognitive function in these mice, indicating that astrocyte clearance of plaques halts neurodegenerative related cognitive decline. Behind the exclusive report, Glory Globe TV, Delhi, Bharat.